Good afternoon. Now it's on. Welcome, members of the Board of Trustees, faculty, students, and honored guests to this 41st LaFollette Lecture. A very special welcome to Gary LaFollette and his son-in-law, Fred Emhart, class of '82. Gary has joined us for almost all of these lectures over the years. Thank you for continuing the legacy of your family here at Wabash. This year, I also want to say thank you to the Wabash Healthy Campus Task Force and to President Feller and Dean McDormand, who worked to bring us back to a nearly normal campus experience and made it possible for us to hold the lecture in person again this year. So thank you. For those of you who are new to the college or attending this lecture for the first time, this annual event is named in honor of Charles D. LaFollette, a graduate of Wabash College and a member of the Board of Trustees from 1952 to 1977. Charles Devon LaFollette, or Leif as he was known by friends, came to Wabash from the small rural community of Thornton in 1916. As a student, he was already actively involved in the humanities and arts, serving as managing editor of The Bachelor, student director of the Glee Club, and one of the student leaders who revitalized the Wabash Players, a theater group on campus at the time. He went on to a distinguished career with the Corning Glass Works and the Dow Corning Corporation. Leif and his wife Ibby generously endowed the Distinguished Professor of Humanities chair as a way to affirm that, quote, "The one indispensable asset and continuing requirement of a good college is superior teaching," end quote. The first lecture in the series goes back to the time of Mr. LaFollette's retirement from the Wabash Board in 1977, when the Humanities Division planned a dinner and formal lecture to honor the lifetime commitment of Mr. LaFollette to the liberal arts and the humanities in particular. It was at the time of Mr. LaFollette's death in 1982 that the Wabash Board of Trustees established this as an annual lecture, always given by a member of the Wabash College faculty. The lecture is one of the ways we continue to celebrate the LaFollette family's generosity, the central place of the humanities in the liberal arts, and the excellence of our teacher-scholars at Wabash College. I hope that after the lecture, you will stay for the reception. This year, we're holding it on the patio in front of the main entrance of the Fine Arts Center, just to your left out the doors, so that we can be outdoors. Let me also ask you at this time to please turn off or silence all your phones and electronic devices. And now I'm pleased to introduce James Makubia, our LaFollette lecturer from 2019, who will introduce this year's speaker.
Ladies and gentlemen, it's my great pleasure to introduce the presenter of the 41st LaFollette Lecture, who is Professor Amanda Ingram. Professor Ingram's LaFollette Lecture today provides a welcome occasion for reminding ourselves about her wide-ranging academic interests and accomplishments. Amanda is originally from Virginia, where she attended the College of William and Mary and double majored in biology and environmental science receiving the Bachelor of Science degree summa cum laude. Thereafter, she completed her PhD in plant biology at Cornell University in New York. Amanda joined the Wabash faculty in 2004. Her main research interests are in plant systematics, which means the study of plant evolution and classification, and she has a long and very impressive list of research projects and subsequent publications. Over the years, Amanda has had an ongoing relationship with the National Science Foundation, starting with an early research fellowship and a dissertation grant, then a research grant in 2009, followed by frequent service on review panels. This led to her being invited to serve over the last 2 years as a program director in the Systematics and Biodiversity Science program at the National Science Foundation. This level of national service included helping to set funding priorities, getting research proposals reviewed, making funding recommendations, and overseeing active awards. Amanda's work at Wabash has included serving as biology departmental chair, and she's currently the division chair of sciences and mathematics. She has also been named the new Treve Professor in Biology, an endowed professorship that provides funding for her ongoing research over the next 5 years. Amanda is no stranger to the humanities. She is an active musician, practicing and playing musical instruments and giving public concert performances, with her favorite instruments being a recorder and an oboe. Working with Professor Sarah Drule in 2017, she also received the Indiana Humanities Quantum Leap Grant, Cultivating Our Gardens. As the title suggests, this grant aimed to support community engagement with botany. Helping people notice the plants around them is one of Amanda's passions and has led her to write a blog about native plants, lead informal botany walks, and help colleagues identify mysterious plants in their own yards and gardens. When Amanda is not teaching biology to her students, she and her husband, Mark, are avid gardeners. Amanda also grows lots of native plants for wildlife. She has cultivated over 100 species of plants on their tiny lot. Wow. Now, to the main focus and attraction of today's 41st LaFollette Lecture, titled, Shaped by History. Please, let's give a warm welcome to Professor Amanda Ingram.
Thank you. Thank you, James, and thank you, Cheryl, for this rather intimidating assignment. For most of the colleagues in this room, explaining how your special field might be viewed through the lens of the humanities would be a piece of cake. But for me, it's not so easy. I'm a scientist, after all, and a botanist to boot. If I'm completely honest, human-focused questions, which are really at the heart of the humanities, Wouldn't rank terribly high in the list of things I find intellectually compelling. Sorry. But as I thought about how I might approach Cheryl's challenging assignment, I began with some careful examination of the questions that do interest me and the approaches I take to answer them. This led naturally to a rather uncomfortable subject that crops up periodically in my special field of systematics. Is what systematists do actually science? So today, I'm going to explore that question, and in doing so, we'll see how my research might relate to the humanities. Now, I imagine all of you have taken a science class at some point, and as a consequence, you probably all know how science is done, by using the scientific method. But in case those memories are dim, or were perhaps muddied by some oral comps that you've sat on, let me remind you of the fundamentals. So a scientist begins by making some sort of observation about the world and uses those observations to generate a hypothesis. A hypothesis is a specific sort of question that ideally is falsifiable or possible to disprove. A scientist then designs an experiment to test that hypothesis. The experiment unfailingly involves controls or unmanipulated conditions. There's also at least one experimental variable that is manipulated in the other conditions. Data are collected and analyzed, and then one evaluates the hypothesis. Was it refuted? Interesting. Was it not refuted? Also interesting. Regardless, the outcome will lead to more questions, maybe something like, why is this so? More observations, more hypotheses, and so on. If you have enough curiosity and time and money, it's a wonderfully self-sustaining process that allows you to probe ever deeper into the inner workings of our world. Let's work through an example of this together. Imagine that you're scrolling through Twitter one day and come across, as one does, the Keeling curve. This is a daily report of the concentration of carbon dioxide in our atmosphere as measured at a Hawaiian research station established by Charles David Keeling in 1958. The interesting observation here is that the atmospheric carbon dioxide concentrations on the y-axis have been rising dramatically over the past half-century. A scientist might consider that observation and wonder how CO2 levels will affect, for example, photosynthesis. As I hope you all know, plants take CO2 from the air and use it to make sugars. One might predict that if more carbon dioxide is available to plants, then the plants might be able to make more sugar. So on the basis of an interesting hypothesis or observation and some prior knowledge, a scientist might formulate a hypothesis. Why rising CO2 should increase photosynthetic rate in plants. Next, the scientist would design an experiment. This would undoubtedly involve including conditions with normal or control CO2 levels and then elevated CO2 levels. If one wanted to design a more elaborate experiment, you might also choose a couple of different kinds of plants to see if they respond uniformly. Maybe you'd choose one of the plants I work on, for example, teff, and then another grain crop like wheat. And these keep jumping forward, and I'm sorry about that. Whew, and now that picture disappeared. Sorry. Okay. So you'd grow both species at both CO2 levels and measure how much photosynthesis they do, fully expecting to find that both do better at higher CO2 levels. You'd collect your data and analyze them, and you know what you would probably find? That wheat does, in fact, do far better at higher CO2 levels, but teff doesn't really improve. So your prediction is partially supported, but how do we explain the species-specific differences? Oh, goodness, I am heavy-handed today. Sorry, folks. Okay. One would probably go back to what is known about the plants. For example, while they're both grasses, they actually use different photosynthetic pathways. Could that explain the difference in response? You'd design a new experiment and continue gaining a deeper understanding about how the world works. It's a marvelously effective and efficient method. Now, as you heard in James's introduction, I am a card-carrying scientist with all of the appropriate degrees and job titles. But the thing is, in my main research activities, I don't actually use this method, or at least not all of it. This is where you may gasp in shock. So what are the questions that interest me, and how do I go about finding answers to them? Am I really a scientist? So as I've mentioned, my scientific subfield is called systematics. It's a relatively young discipline, one that wasn't possible to imagine until we had a good handle on how evolution works. But it has much deeper roots in an older field called taxonomy, which is focused on assigning names to the organisms around us and building classifications to organize the planet's vast biodiversity. I would argue, as many have done before me, that taxonomy is an instinctive activity for humans. For one thing, humans like to talk, and it's hard to talk about things if you don't have a name for them. As an example, we could refer to some of the trees in our arboretum as large trees with simple leaves that have rounded lobes and that make oval-shaped acorns that are about 3/4 of an inch long and have a warty cap and that are reasonably palatable if you know how to prepare them. But it's far clearer to call them white oaks, or even better, Quercus alba. All of those facts about the trees are, in a way, embedded in the name, at least to those who have taken the trouble to learn about them. So practicing taxonomy by naming things provides shortcuts to facilitate accurate and efficient communication. But names alone aren't always enough to help us cope with the vast and complex world we live in. So humans also have an inherent tendency to develop classification systems to organize this information. To stick with our tree example, you might notice that there are actually several kinds of trees out in the arboretum that make acorns. Some of them may have unlobed leaves or pointy lobes, and the acorns might be different shapes, but they are undeniably more similar to each other than they are to any of the other fruits that you see in Indiana. So we would group these together as oaks, or members of the genus Quercus, and if you're really sophisticated, use adjectives like alba or rubra, or if you prefer, white or red, to differentiate among them. But even if you can't distinguish the various kinds of oaks, just knowing that they are oaks tells you a great deal. They're trees, their wood is probably useful, and they make fruits that are acorns. So classification and taxonomy work together to facilitate communication, organize the vast biodiversity surrounding us, and even help us make predictions about the species' traits. Classification and taxonomy have been happening for as long as humans have existed, and possibly before. But naming and classifying really came into their own as rigorous pursuits with the father of modern taxonomy, Carl Linnaeus. Linnaeus established many of the conventions that we continue to use today in naming organisms in his influential publications, including Species Plantarum, which was published in 1753. Linnaeus is credited with formalizing the Latinized binomial, as you can see here in a page from the text. So this is a page for the genus Quercus. You can see the generic name here, and then off to the left is an adjective describing a particular species, the specific epithet. His classification was based on floral characters, as you can also see at the header of this page, so I won't go into all of the details, but polyandria, for example, refers to the fact that these plants have flowers with many stamens, male parts, on them. As an aside, the classification system was seen by some as quite scandalous. As you can see, Linnaeus called it the sexual system because it's based largely on these reproductive characters, and he had the temerity to call hermaphrodite flowers perfect and unisexual flowers imperfect. This did not sit well with some. One critic called the system loathsome harlotry. But it turns out to be effective in its ease of application, and many of the categories that Linnaeus identified are still in use today. Though to be fair, Linnaeus also described a group that included, among other things, some grasses, some sedges, and maple trees. Even the least botanically inclined upon you might conclude that that is a far less intuitive group than, say, Quercus. Linnaeus' system arrived at an opportune moment, at least viewed from the perspective of Eurocentric assessment of scientific progress. Around the time that Linnaeus developed his taxonomic conventions and his classification system, European naturalists were increasingly traveling the world and discovering species not yet known to Western science. Linnaeus' system allowed names to be applied and enabled rapid categorization of, and thus communication about, the incredible biodiversity found throughout the world. But before we move on, I want to take a moment to acknowledge some of the troubling ways scientists in my field typically frame our exploration of the natural world. Let's start by returning to our friend, the white oak. Its full name is actually Quercus alba L. That L at the end is called the authority, and it's essentially a citation of the person who named the species, giving that individual credit for discovering it. L is our abbreviation for Linnaeus, and many species names bear his authority. But did Linnaeus actually discover white oaks? Of course not. For one thing, the species is restricted to North America, and Linnaeus never visited this continent. He sent plenty of his students to explore North America, many of whom suffered tremendously or even died on these expeditions, but even they were not the first humans to know these plants. Indigenous peoples already knew and had named this species. But because an enterprising European guy had access to vast collections of plants from all over the world and access to publishing to disseminate his work, he continues to receive the credit for discovering and naming thousands of species. This is just the tip of the iceberg of the complex and often disturbing history of colonialism in my field. But back to taxonomy and classification. Does what I've described so far sound like science? There's certainly a great deal of observation that goes into putting names on things and putting them into groups. You need to understand the species' physical characteristics and how they compare to other known species to figure out if there are different species and with whom you might group them. So you could argue there's data analysis. But are there hypotheses? No, not really. And are there manipulative experiments? No. But I hope you're all thinking, surely this practice has changed since 1753. And of course you would be right. In 1859, Charles Darwin published a figure and a lengthy explanation of it in The Origin of Species that eventually changed everything for taxonomy and classification. This unassuming collection of lines elegantly illustrated Darwin's radical idea that all species that live and have lived on our planet could trace their origins to a single common ancestor, and that the biodiversity we see today arose as a consequence of billions of years of selection on naturally existing variants, as well as some other evolutionary processes that came to be understood later. So how did I get all of that from this diagram? First, Darwin intended for the lines, or branches as I will call them, to represent lineages. The nodes where those lines intersect represent common ancestors of two or more lineages. The top of the diagram is the present, the bottom is the past. Branches that peter out before the present represent extinct lineages, like these, which may or may not be represented in the fossil record. Darwin envisioned the ends of those branches as representing individual species and multiple genera, so we might label them oaks and chestnuts. There are only a handful of species represented here, but in theory, we could extrapolate out to all known species, connect the lineages, and trace them back to the ancestor of all living species. After setting out his theory for how evolution by natural selection works, which is generally the part of this treatise that gets all of the attention, Darwin went on to outline what I find even more interesting: the implications of his theory and this diagram for classification. By the 1850s, there was increasing appetite for classifications to mean something and to be more than a convenient way to organize biodiversity. In other words, the naturalists of the time were growing dissatisfied with so-called artificial classifications like Linnaeus' sexual system, which, after all, produced that group that lumped together maples and grasses. Instead, they sought to produce what they called natural systems of classification. Some naturalists at the time felt that natural systems should reveal the plan of the creator. Darwin rejected this idea and instead returned to an idea put forth by Linnaeus. Quote, namely, that the characters do not make the genus, but that the genus gives the characters, end quote. Darwin went on to say, quote, that community of descent, the one known cause of close similarity in organic beings, is the bond, which, though observed by various degrees of modification, is partially revealed to us by our classifications, end quote. In other words, if we get it right, our classifications should reveal something about the relatedness of taxa, and this is because groups share features because they share a common ancestry. For example, we put species in the genus Quercus because they make acorns. The genus gave us that character because the genus Quercus is a natural entity, not a human construct. Oaks make acorns because their common ancestor made acorns, and the character was passed down through natural processes to our present-day species. A natural classification might then allow us to predict that all oaks share a more recent common ancestor than any oak does with a chestnut, and we can identify those most recent common ancestors of groups of species in the diagram. So for the oaks, the most recent common ancestor would be right here, and if you look for the most recent common ancestor of all oaks and chestnuts, you'd trace those branches back to where they intersect down here. This was a tremendous insight. Now classifications can be viewed as hypotheses of properties of nature. Rather than representing groups of species that are simply more similar to each other than they are to other equivalent groups, as is the case with Linnaeus's artificial system of classification, taxa now can be viewed as hypotheses of common descent. Natural taxa came to be thought of as groups that could be traced back to a single common ancestor. So when Darwin's ideas are realized, classifications move beyond being a simple filing system and instead represent the underlying biological processes that generated the diversity of life. So, our taxonomy and classification science now. We still have the observation piece, and now I've introduced the idea that classifications represent hypotheses explaining biological phenomena. But there are still no experiments, and I haven't really explained how Darwin came up with this branching diagram. In other words, what were his data and how did he analyze them? The truth is, this diagram you're looking at is really a thought experiment based on Darwin's extensive observations of organisms and his hypothesis about how species originated and diversified. The letters don't represent actual taxa, there were no data, and no algorithm was used to construct the tree. But over the ensuing decades, taxonomists began building diagrams representing evolutionary histories that did incorporate real taxa and real data. Among them, this figure, produced by Charles Bessey in the late 19th century. Known fondly as Bessey's cactus, this diagram is a culmination of Bessey's encyclopedic knowledge of the flowering plants and his attempts to arrange taxonomic orders by their advancement. Each pad of that cactus represents an order. The groups at the bottom are characterized by what Bessey thought of as more primitive characters, while those toward the top are more advanced. Even though this diagram depicts real data assembled from real taxa, there are two serious issues with it and with many other similar classifications developed during the late 19th and early 20th centuries. The first problem is Bessey's focus on distinguishing primitive and advanced groups. This was wildly misguided. A clear implication of Darwin's theory was that all living species descended from a common ancestor. All living species, therefore, ultimately belong to the same ancient lineage, and we all possess characters that are the result of billions of years of evolution. None of us is really more advanced than any other. But this has been a challenging idea for people to accept, and vestiges of this mindset linger in my field even today. We humans are accustomed to thinking of ourselves as special and somehow better than all other living species, and we often frame evolutionary trajectories accordingly. The second problem with early evolutionary trees like Bessey's was that the method for arranging taxa was entirely subjective. As these hypotheses of evolutionary relationships proliferated, systematists became concerned with how to evaluate the merits of competing hypotheses. Was there a repeatable method underlying the generation of these diagrams? There wasn't. So how do we evaluate ideas based on these subjective approaches? When philosopher Karl Popper introduced the idea that hypotheses should be falsifiable, systematists began to wonder if their hypotheses met this mark. They did not. Interest grew in developing more objective methods for producing and testing falsifiable hypotheses of evolutionary relationships, which might in turn form the basis of classification schemes. These methods needed to have a firm philosophical grounding, and they needed to be applicable to the wide array of data that a systematist might want to incorporate. Enter Willi Hennig. Hennig was a German entomologist who published his most influential work, Phylogenetic Systematics, in 1950. The text was translated to English in 1966 and quickly became a lightning rod for controversy and debate, sometimes friendly but often vitriolic, that has continued into this century. One of Hennig's key contributions was to set the stage for developing an objective, repeatable method for constructing these trees based on something more meaningful than perceptions of overall similarity. Since we can't directly observe what happened in the past, he and other like-minded systematists who followed advocated for relying on the principle of parsimony, or Occam's razor. The simplest explanation is the preferred explanation. In this case, that meant building a tree that required the fewest evolutionary changes to represent what we know about organisms and how their traits have changed over time. This enabled the characters, inherent and natural features of organisms, and a simple optimality criterion, minimizing evolutionary steps, to work together to produce trees on which we could base or evaluate classifications. Hennig also refined our understanding of what natural groups should be. In addition to all members of a taxon tracing their evolutionary history back to a single common ancestor, Hennig felt strongly that a natural group should include all descendants of that common ancestor. This may sound uncontroversial, but it called into question the validity of many familiar taxa, a concept that can be illustrated with the flowering plants. Many of you have probably learned that there are two kinds of flowering plants, monocots and dicots. The distinction comes from how many cotyledons, or embryonic leaves, they have. So you can see two on the left for a dicot, one on the right for a monocot. One of the things we've learned in the past few decades is that this distinction may be obvious and useful, but it doesn't accurately define natural groups. In this evolutionary tree of the flowering plants, groups highlighted in green have two cotyledons, and those in yellow have one. As you can see, the monocots all share a common ancestor, back here, you trace back, and no dicots are derived from that ancestor. Monocots are a natural group according to Hennig's criteria. But if you trace back to the common ancestor of all dicots, which would be way back here, you see that some of the descendants of that ancestor have one cotyledon. Dicots do not meet Hennig's criteria for natural groups. The names monocot and dicot aren't equivalent. The former describes a complete lineage, whereas the latter is incomplete and therefore unnatural. Together, these ideas led to rigorous methods for tree construction that were gradually adopted by the community, as well as a more nuanced understanding of how to define natural groups that may form the basis of classifications. The stronger conceptual framework, paired with technological advances in computing and DNA sequencing, has enabled systematists to generate and analyze data sets that include large numbers of taxa and an enormous number of characters. The systematics community continues to argue about exactly which data and which methods of analysis are best, but the one thing we can probably agree on is that we've come a long way from diagrams like Bessie's cactus, which were based on opinion. Now we have the ability to produce trees, which we've come to call phylogenies, that are far more accurate, with methods that are far more objective. These phylogenies have enabled some giant leaps forward in our understanding of how life has evolved. For example, some groups that I suspect will sound familiar to all of you, invertebrates, fish, reptiles, are now known to be unnatural groups that fail to include all descendants of their most recent common ancestor. Vertebrates arose from within the lineage that includes all of the animals without backbones. Tetrapods, four-legged things, arose out of the lineage that includes all of the fishes. Birds are reptiles. This has reframed the way we think about the history of these groups. Phylogenies can provide insights beyond assessments of the hypotheses represented by our classifications too. We can put dates on the nodes, either by working backward based on the rate at which DNA typically evolves, or by calibrating with fossils. This can help us understand what the world was like when the ancestors of living groups existed. Where were the continents? What was the climate like? This in turn can help us understand how taxa have dispersed all over the world and how characters have evolved. Phylogenies can also be used to understand more recent questions of evolutionary origins. Curious about where SARS-CoV-2 came from? Sample the sequences of coronaviruses in a bunch of wild animals, build a phylogeny, and determine which host animal has coronaviruses sharing a most recent common ancestor with our current plague. So let's get back to my question. Is what contemporary systematists do science? We make observations about living creatures, we develop hypotheses about how they might be related, and we collect large quantities of data. We analyze those data with mathematically complex algorithms to generate phylogenies, which allow us to evaluate our hypotheses and ask more questions. Yet we never do any manipulative experiments. And this is because what we're fundamentally trying to do is to understand the history of our organisms. What did extinct ancestors of the species we now know look like? How did these species diverge? How have they spread around the world? How did new characteristics evolve? All of these are questions about the past, whether quite recent or unfathomably ancient. So I would argue that we are, at heart, historians in lab coats. We're trying to make sense of the outcomes of nature's experiments rather than experiments of our own design. Unlike humanities scholars who study history, most systematists have little interest in humans and instead spend our lives occupied with the obscure, underappreciated organisms of the world. But in many ways, our activities mirror those of historians. We rely on archives of information, such as DNA sequences preserved in our genomes, to draw inferences about what happened in the past. As is true for historians, our data sets are often incomplete. In the case of the evolutionary process, many lineages go extinct and leave no trace of their existence, just as many conversations, letters, or other artifacts that might shed light on human history never make their way to an archive. And our data sets may even be misleading, thanks to the strong forces of nature shaping the species that have survived, mirroring the way people's biases and selectivity affect the information they leave behind. But we do our best to use these wildly incomplete data to form an understanding of our past. Sure, we apply scientific laboratory techniques and mathematically complex analytical methods to collect and make sense of our data, and we generate and test hypotheses. So, begging Stephen Colbert's forgiveness for this coinage, you might say our work has some science-iness to it. But at its heart, one could make the case that systematics is a historical exercise. So I think this is where we can see how my scholarly work relates to the humanities. And Cheryl Hughes's LaFollette lecture, titled Integrity, come on, thank you. She wrote, "Humanistic studies give us insight into ourselves, our values and ideals, and our development over time." I submit that systematics gives us insight into nature and what nature values and the development of biodiversity over time. So what aspects of nature interest me, and how do I go about understanding what nature values? First and foremost, I am a plant systematist. I could give another entire lecture on why I'm so fascinated with plants, but for today's purposes, let's just say that I love that plants aren't bound by many of the rules that seem to limit the way animals exist in the world. Two rule-defying attributes that are particularly interesting to me are hybridization and polyploidy. Plants are notoriously promiscuous, happily mating with other species on a regular basis, producing offspring that are often quite viable and even fertile. This is often associated with polyploidy, or having more than 2 sets of chromosomes. Polyploidy turns out to be extraordinarily common in plants. Somewhere around 70% of living plant species are some sort of polyploid. We don't fully understand why plants don't mind having extra chromosomes or why they're able to mate so freely, but we do know that all of that extra genetic material put together in new combinations is terrific fodder for evolution, and understanding the historical patterns of these processes is a first step toward understanding how and why polyploidy and hybridization influence plant evolution. I'm also motivated to better understand the histories of human-plant interactions. So these complementary interests in economically important plants and polyploidy are what brought me to the group I've spent most of my career working on, the genus Aragrostis, commonly known as the love grasses. The most well-known economically important species in the genus is Aragrostis teff, commonly known as teff, a cereal crop grown in Ethiopia and used to make breads like injera. Teff seems like a wildly improbable crop. Its grains are tiny, and it's prone to flopping over or dropping its fruits at inopportune times. But it thrives in the challenging conditions in Ethiopia, tolerating heavy soils, unreliable rains, and wild elevational variability, making it an essential crop for food security in this low-income nation. Several other Aerogrostis species provide forage for livestock, and others are nasty weeds important for their negative economic impacts. But most of the approximately 400 species of Aerogrostis are little wild grasses about which relatively little is known. They can be short-lived annuals or long-lived perennials. If you look closely enough, you'll find they exhibit striking morphological variation, especially in their floral structures. They grow all over the world and on a variety of soils, but they're most diverse in the tropics and subtropics and in disturbed areas or sites where they might experience some water stress. This is likely due to the fact that they use a modified form of photosynthesis called C4 photosynthesis, which enables them to conserve water and continue cranking out sugars to build their bodies and fill their fruits, because yes, grains are fruits, even when water is limiting and light levels are high. I started working on Aerogrostis largely because I was curious to know what wild species gave rise to teff. Knowing this could facilitate improvement of the domesticated species because wild relatives often harbor desirable traits that can be bred into the domesticate by hybridization and selective breeding. Comparisons with wild relatives also provide key insights into how human interactions shaped the history of crop plants. I found teff's closest wild relative easily enough, but on examination of the phylogenies that addressed this question, it became obvious that some more fundamental work on classification was necessary. So in this phylogeny, you can see that most of the species named Aerogrostis, they're highlighted in this sort of orangey color, can trace their origins to a single common ancestor, where this gray star is. But other species fall outside that lineage, these guys, for example. There are also a handful of species referred to as segregates who have at times been placed in Aerogrostis and at others have been moved to their own genera. Some of those are in the main Aerogrostis lineage. You can see this yellow species right here. Others are outside of it. This phylogeny also reveals that not all descendants of the common ancestor of most Aerogrostis species are named Aerogrostis, so we have some things that have no highlighting on them, for example. As a consequence, one of the major questions that has occupied me in the research lab is how Aerogrostis should be circumscribed. The work I'll tell you about today has been focused on improving my sampling of key Aerogrostis species, sequencing some genes, and building phylogenies like this to understand the group's history and to test the hypotheses represented by these names. Before I show you some of the things my collaborators and I have learned, you need to know how past botanists have decided to place species in Aerogrostis. The first official mention of Aerogrostis was actually on the previous slide, which maybe we'll get to see. Forget it. Okay, it was actually in Linnaeus' Species Plantarum. But he applied the name as a specific epithet, the species name, rather than as a generic name. But in 1812, just 20 short years before some Presbyterians kneeled in the snow and founded this college, a French botanist named Palissot de Beauvoir correctly realized that the species to which Linnaeus applied this name did not actually belong in the genus to which he had ascribed them, and instead elevated the name to the rank of genus. His description of Aerogrostis, which you can see on the screen, if you can read Latin, then you're in good shape, lists several characters by which one might recognize a grass that belongs in this genus, including 2 characters related to the organization of the flowers. First, in grasses, flowers are organized in structures called spikelets. So this is a spikelet. In Aerogrostis, those spikelets contain many flowers. Each one of these little leafy things contains a flower inside. Additionally, those spikelets are arranged in an inflorescence type called a panicle, a structure with multiple levels of branching, which you can see here. So here's a main axis, there's one level of branching, and there's another. A few other characters have come to be recognized as important in placing species in Aerogrostis in subsequent years, including having 3 veins on leafy structures in the spikelet called the lemma. And you can see those veins here, those dark green lines. If we view this through the lens of modern systematics, we effectively have hypotheses of characters present in the common ancestor of all extant species of Aragrostis. In other words, this name represents a history, and my job is to deepen our understanding of that history. So let's look at a few examples to illustrate how these plants' appearances seem to have misled us about their history and what my collaborators and I have done about it. The first trickster that we'll talk about is a species I encountered when on a collecting trip through the southwestern United States and Mexico with some colleagues from the California Botanical Garden. One of them, Hester Bell, was working on a grass called Disticus spicata, which you can see here. So we made lots of stops in the places it grows, which are salt flats or playas, like this one, to collect them. On one such day, we happened to grab something that superficially looked like Disticus, but on closer inspection turned out to be a species named Aragrostis obtusiflora. It was a surprise to me to find it there. Aragrostis species aren't common in soils that are quite this alkaline, and its non-reproductive parts were a bit unusual. It had leaf apices that can puncture skin, a creeping habit, and, as we discovered later in the lab, atypical leaf anatomy and microhairs. Despite that, it had all the usual reproductive features that cause botanists to put species in Aragrostis. We were excited to see what the DNA told us, and sure enough, the sequences revealed that the reproductive features were misleading us. Aragrostis obtusiflora is, in fact, quite distantly related from other Aragrostis species, and I hope to show you that. There it is. Right here is Aragrostis obtusiflora. The rest of the genus is up here. So we gave it a new generic name, Kalinia, derived from the Arabic for alkaline, so that our classification better reflected the evolutionary history. A different sort of story can be told about some grasses in the genus Ectrosia, which I first met in Australia. I was keen to see these plants in person because some previous phylogenetic analyses of the subfamily to which Aragrostis belongs had suggested that Ectrosia was closely related to Aragrostis, but the results were preliminary because neither genus had been sampled very thoroughly. Ectrosia differs from Aragrostis in several ways, largely related to the reproductive structures, as you can see on the screen. At a glance, they look almost nothing alike, but the differences are actually quite simple. Ectrosia doesn't have as many flowers in each spikelet as Aragrostis. The flowers are more spaced out in Ectrosia, and the lemmas in Ectrosia spikelets have long awns or projections, like you can see here. The last feature is quite striking and makes our Ectrosia species quite distinctive. But again, the DNA sequences help us understand how much weight to give those simple morphological changes. The phylogenetic analysis reveals that Ectrosia is nested within Aragrostis. So here's all of Aragrostis. I've zoomed in on the part in the box. And here's Ectrosia nested within the other genus. So it appears that Ectrosia evolved in Australia from some Aragrostis species. They're really just gussied up Aragrostis. And the awns are probably a simple adaptation to help the seeds disperse. They've been renamed. Now let's examine one final case, Aragrostis walteri. If a grass that is narrowly distributed in Namibia and that has absolutely no economic importance can be famous, Aragrostis walteri would qualify. This is a species that was originally described by Pilger in 1941. He placed it in Aragrostis because the multi-flowered spikelets, which you can see on the right, look like those of other Aragrostis species. The plants also have paniculate inflorescences, as you can see in the photo of the whole plant. A phylogenetic analysis of morphological data in the mid-90s confirmed this placement. But in 1984, a South African botanist named Ellis made the shocking discovery that Aragrostis walteri deviates in one striking way from all other Aragrostis species. It has leaf anatomy typical of grasses that use normal or C3 photosynthesis. I didn't hear any of you gasp, so you probably need some context to understand why this was so shocking. So first, let's talk briefly about how these two forms of photosynthesis differ. C3, or normal photosynthesis, occurs within a single cell. CO2 diffuses into the cell, is, good grief. Okay, the CO2 diffuses into the cell, is bound to the most abundant enzyme on the planet, called rubisco, and is converted into sugars. That little green guy's a sugar. This works well for many species, but there's a slight hitch. Rubisco is a terrible enzyme. It's terrible because it can't tell the difference between oxygen and CO2. So sometimes, there we go, sometimes oxygen binds to it instead of CO2. This is inefficient, and it also generates a byproduct that has to be destroyed by the cell. Most plants carry on despite this inefficiency, but the problem becomes worse when plants live in certain types of marginal habitats, such as hot or water-limited places, and this inefficiency ultimately becomes untenable. Happily, a number of solutions to deal with rubisco's shortcomings have evolved. One that has evolved dozens of times in the land plants is C4 photosynthesis. Plants that use this pathway basically don't give rubisco the opportunity to bind with oxygen by artificially increasing the CO2 concentration where rubisco is present. They do this by altering a few things about their anatomy. So this is an Eragrostis species, a little slice through its leaf. And first, rubisco is present only in certain leaf cells. These cells near the vascular tissue called the bundle sheath cells. These are interior cells that are buffered from atmospheric gases by the surrounding mesophyll cells. So CO2 diffuses in from the outside into these mesophyll cells, and when there, it is bound to a 3-carbon molecule, producing a 4-carbon molecule, hence the name C4. And this 4-carbon molecule is then shuttled to the cells where rubisco lives. When it gets there, CO2 is released, giving rubisco lots of things to work with, and sugar is made. This isn't an energetically expensive process, but it allows plants to thrive in places where they wouldn't otherwise. It also requires a huge number of changes that I would love to show you. There we go. First, veins have to be closely spaced. So in this C4 leaf slice, you can see the veins are here and here. They're very close together, just a couple of cells in between them. Whereas in this leaf from Eragrostis walteri, here's a vein, and you can see there are at least 10 cells in between those two veins. The veins have to be close together in C4 plants so that no mesophyll cells are very far from the cells that have rubisco. Additionally, a bunch of proteins have to be localized in a very specific way, and many of them are in much higher abundance than they are in a C3 plant. But as I said, this suite of characters has evolved repeatedly in the land plants, including at least 20 times in the grass family. But what's really interesting about this is that phylogenies tell us that more or less all of those evolutionary transitions in photosynthetic pathway appear to have been in one direction, from C3 photosynthesis as the ancestral condition to C4 photosynthesis as the derived condition. Reversions from C4 to C3 photosynthesis were thought to be impossible, which, if you think about it, is fascinating. So much does seem possible, given the vast timescales on which evolutionary change occurs. So it's a bold claim to suggest that a change is irreversible. But then came this revelation that Eragrostis walteri has anatomy typical of a C3 species. All other Eragrostis species are C4, and in fact, all the species in the subfamily to which Eragrostis belongs are C4. So if Eragrostis is a natural group, then Eragrostis walteri must have reverted to C3 photosynthesis from a C4 ancestor. In other words, Eragrostis walteri appeared to have escaped the constraints of its history. Hopefully now you can understand why Eragrostis walteri was famous, at least in certain circles. Well, some colleagues and I decided to test the hypothesis that this species name represented, so we got our hands on some material. First, we needed to confirm that Eragrostis walteri does in fact use C3 photosynthesis by conducting analyses of isotopic composition. It does. Then we sequenced some DNA to confirm that its morphology wasn't tricking us, and that it actually was a descendant of the most recent common ancestor of all Eragrostis species. When we got the sequences back, it was immediately obvious that the morphology had, in fact, tricked Pilger and all of the subsequent agrostologists who studied this species. Its sequence was highly divergent from other Eragrostis species, and a phylogenetic analysis of a broad sample of grasses, there we go, revealed that it is actually related to a large group of C3 species. So C3 is coded in yellow. Here's Eragrostis walteri, lots of other C3 things around it. Eragrostis would be here in this orangey red triangle. So what did we learn? There are certain evolutionary paths that still look extremely difficult, and certain histories may be impossible to escape. Historical constraints set by nature's values are real. But it also illustrates how important it is to look carefully and to try not to dismiss data that are inconsistent with your initial hypothesis. When I finally had a chance to meet Eragrostis walteri in person, it actually made no sense to me that Pilger had put it in Eragrostis in the first place. First, it grows in a stream. Eragrostis hardly ever does that, and certainly not in a mucky stream like this one. Second, if you look at those lemmas closely, there are actually 5 veins, not 3, as we see in the rest of the genus. And those lemmas, as you can see in the photo on the left, also have pointy ends. So this is also not typical of the genus, though not impossible given what we've seen from Ectrosia. So there were clues to the species' actual history all along, but then hindsight is 20/20. Well, I've elaborated on several examples where initial hypotheses of relationships, as represented by taxonomy, were inaccurate. I should point out that systematists have actually done pretty well overall. Most of the Eragrostis species I've sampled do fall into a single natural group, and with additional sampling, some taxonomic changes, and some more data, I'll be able to make some inferences about the group's history, where the genus originated, what the most recent common ancestor may have looked like, how polyploidy has influenced diversification in the group, and so on. These phylogenies can have practical value too. For example, one of the challenges with growing teff is that its grains sometimes fall off before they can be harvested. But fruit dehiscence, as this is called, varies quite a bit in the genus, so some colleagues and I are using the historical framework that phylogenies provide to understand how this trait has evolved, what genes are involved, and how we might imagine and affect a future in which teff is a more efficient crop. I hope you've learned today that systematics is a field whose roots were in organizing and naming life, but the field has evolved to begin probing the historical patterns that our taxonomies can reflect. We infer these patterns by looking at a snapshot of time, and by doing so, we can see what worked, what nature valued, how history shaped life, and what constraints biology put on evolution. And this historical perspective allows us to consider how our own actions may shape the future of our planet's biodiversity. We know, for example, that atmospheric CO2 concentrations have fluctuated in the past, but they've never risen at this pace. Systematists, with our sense of the extreme age of our planet and the vast expanses of time required for lineages to adapt to changing environments, understand that we're not giving other species the time they need to adjust to the new normal. Our understanding of the organisms we devote our careers to and their pasts can also help us predict who will win and who will lose in a new nature. Those C4 grasses that are so well adapted to drought and heat, conditions we know we're likely to see more of in the future, they evolved at a time when atmospheric CO2 had dropped dramatically. They'll lose one of their most important competitive advantages in a future where the habitats that can support them will almost certainly become more abundant. And as the phylogenies have told us, it is exceedingly unlikely that they'll revert to a form of photosynthesis that can take advantage of the new conditions. Who will take these grasses' place? Will any species be able to take their place? What will happen to Ethiopians who rely on a C4 crop for food security? So this is why systematists spend our time exploring the consequences of nature's experiments. By understanding what shaped our past and what nature valued, we can prepare for our future. Thank you.